I am glad to, well, not see you today. I actually miss y'all very much, and I hope everything's going okay. I still have a cough, and my voice may go in and out on me. Yesterday, you did two things. You watched a video from the Amoeba sisters, and also there was a CK12 assignment I told you to take notes on. So I just wanted to verify a few little things with you to make sure that you had them written down in one form or fashion. So from the Amoeba sisters, we, make sure that you understand changes occur, mutations occur not only in the nucleic acids of animals, but also in viruses, and of course we're seeing that right now. Many of them are neutral, so some of them, depending on what happens, don't affect you at all. You've got some that are harmful and some that are helpful. Remember, the lactose tolerance that we have been looking at, that was helpful, and that was a mutation that occurred. Okay, so this was something that was actually beneficial to many people. Not everybody is lactose tolerant. Okay, and also remember, they are random. We have this concept in our head that if we don't get this out of our head, it'll be difficult for us whenever we start on this whole evolution thing, which is coming up. We have this concept that some organism wants to be a certain way, and so it can kind of will itself to be a certain way. That's not what it is at all. These things are random. And then you've got different factors that cause them, and depending on where they cause them, that could be passed on to an offspring or possibly not passed on to an offspring. Okay, so this video does a good job to explain some different types of mutations, and they do a pretty good job in explaining that your substitutions, your insertions and deletions, these can be especially dangerous, and I'm going to go more over that in a moment. But the reason these are especially dangerous is because they give you completely different amino acids. They're frameshift mutations. Okay, so I'm going to cover this a little bit more. In the Amoeba Sisters video, they describe that you've got different types of chromosomal mutations. Okay, some of them are duplications. I'm going to describe that later. Deletion, inversion, and translocations. The most vulnerable times are whenever you have DNA replication. So this is a pretty vulnerable time right now whenever your DNA is replicating. A lot of things can go wrong. And then also meiosis. That's a pretty vulnerable time for chromosomal mutations. Okay, non-disjunction. This happens, there's a better picture of this that you're going to see later on, but this happens when in the cell dividing, something does not separate. Okay, so disjunction would be separate, and non-disjunction would be to not separate. Okay, this is going to be pretty bad because it results in having either too many chromosomes or not enough chromosomes. We're going to see some examples later. Alright, now mutations. Some of them can be passed along, some of them can't be passed along. If they are in the sperm of the egg cells, and you'll see later on, these are our germline mutations. These can be passed along to offspring. But if it's in a somatic cell, those cannot be passed along to the offspring. Some substitute, we're going to see a lot about sickle cell anemia. So they did a real quick little synopsis on what sickle cell anemia was in the video that you watched. I'm going to emphasize that people who that are carriers for sickle cell anemia, they have a protective factor against malaria. Okay, so the people who just carry the gene but don't actually have, you know, it's a recessive gene, and we're going to learn, you know, more about the recessive and stuff, but I think you remember that from when you were in 8th grade, whatever grade it was you took this, 7th grade, life science. You have dominant and you have recessive, and we're going to learn more about this too. But if a person can be a carrier, and if you are a carrier, this is helpful to you because you can be protected a little bit better against sickle cell anemia. Okay, so that was your kind of key points from the video that you watched about mutations from the Amoeba sisters. Love them. You also had a reading to do. This was from your CK12 assignment. We're going to have a lot of stuff from CK12, so make sure you get on there. They talked about, and I thought that this was pretty cool, albinism. This is when, like albinos, if you go over here to Flint River Aquarium, there is an albino alligator, an albino alligator. They showed a picture of an albino alligator, and I don't know if it was from Flint River Aquarium or not, but we do have one over there. Okay, now they used a little bit higher terminology, which actually matches your academic lexicon in our vocabulary, or excuse me, in our standards. Okay, so they talked about the difference between germline mutations. These occur in the gametes. These are passed on to offspring because they are in the sex cells. Okay, remember somatic, some means body. If they are in somatic cells, these are in a person's body, and they can't be passed on to offspring because they're not in the egg or the sperm cell. So for example, an example of a somatic cell mutation could be, you know, something that gives me cancer. So just because maybe an adult gets cancer at 50 whatever years old, that doesn't mean that the that trait is passed on to the offspring. Maybe I'm out smoking, drinking, not using sunscreen. Well, that doesn't mean that my child is automatically going to inherit my crazy, wild side. Let's hope not anyway. Okay, now, here's where you need to be able to interpret pictures. So I wanted to go through this again with you because they had some pictures, but I felt like I could, you know, kind of help clarify this, because you're going to see pictures whenever you take this, you know, these quizzes or these tests. Okay, deletion means to just take something away. So if this is my original gene, notice, so I've got my brown, my gray, but my blue is deleted into my copied gene. Okay, so something happened, something wasn't right, and it deleted this. Sorry, y'all, I've still got a bad cough. Okay, so now if it deletes this, this is one of those that they refer to as like a frameshift mutation, because what it does, it can totally take away an amino acid that is put together, and the protein will not be made correctly. Okay, deletion, taking something away. So if you look at a picture and you see that that is taken away, that's a deletion. Okay, duplication. Again, this is a pretty bad thing because it's a frameshift type of mutation, and it causes something totally different to be, you know, in terms of an amino acid to be made, a different amino acid. So what's happened here is, instead of, you know, our original, we've got one extra brown gene duplicated here. Okay, so instead of going brown, gray, blue, green, we've got brown, brown, gray, blue, green. Okay, so this is duplication. Again, a frame shift because it allows one more gene to be there, and that will copy a wrong amino acid, a wrong protein. Okay. Now inversion. If you invert something, you flip it. Okay, you flip it in some way. So if our original here again, brown, gray, blue, green, we'll notice that our blue and our gray are flipped. Okay, that is an inversion. Okay, an insertion. This is where we insert something. Okay, so now again, here's our original. And we've got our brown, brown, gray, gray, blue, blue. But then, whoa, what happened here? We've got a light green inserted. And then our dark green. Okay, so our copy of that gene is, there's a shift that's going on. We've got, because of this insertion here, we've got one more amino acid or something that's not going to be right. Okay, so I wanted to show you these in your different colors because I felt like they were a little bit easier to see with different colors than they were in that CK12 reading that you were doing. Okay, now translocation. This was one that was kind of weird to me, so I wanted to try to explain it a little bit better to you. Okay, but if you've got translocation, these are where you have, you know, your original genes. And when your copies are made of these genes, so we've got a brown, a gray, and then we've got the blue and green. These are our originals. And this is another gene right here where we've got a light green and a pink. Okay, with a translocation, trans means across, so it's going to a different place, a different location. Notice that this green, instead of copying, you know, an original gene, brown, gray, blue, green, it copies brown, gray, and this one right here. Well, then this one is going to go and take the wrong position too. So this one goes here. So if you see something that, you know, if you see a picture where you've got two different colors that just end up in the wrong places, like these have, okay, see these two went together originally, and these are the original 4 we've been working with, and then the ones that we get are just nothing like that. They've gone across. This is translocation. Okay. Now, we've got, I do like the way that the CK12 talked about point mutations and some different frameshift mutations. Things that I felt like they did a pretty good job in explaining this. But a point mutation is a change in a single nucleotide. Okay, they can be silent. And when we say silent, that means no effect. And if you look at the little chart where we code for our amino acids, we have this built-in, this is a built-in dummy-proof thing that God has blessed us with, or you know, that whoever's blessed us with, if you're not a if you don't believe that God blessed us with these things. Okay, but this is sort of a built-in dummy-proof way, because there are so many ways to make mistakes. Now, there are multiple things, multiple codons that code for amino acids. Like CAA codes for glutamine. Well, also CAG codes for glutamine. Now, if you have a point mutation where you have, instead of a CAA, you end up, for whatever reason, your transfer RNA brings a G instead of an A, well, we got lucky. Both of these code for glutamine, so since we're getting the same amino acid, we're not going to see any effect. Okay, that is called silent. That is a silent point mutation. These do happen, and again, this is excuse me, a way for us to have some mutations that are not causing actual problems. Now, whenever we have some of these missense or nonsense frameshift mutations, these things are difficult for us. And the what you read yesterday, it did talk about this. There was a little bitty section on these missense. There was a table there. So I wanted to explain just a little bit more. I didn't feel like this was made as much sense as it could. But now, if you have a missense mutation, a change, like for example, if our original amino acid, our codon, our original codon, gave us CAA, which is glutamine, if for some reason the tRNA brought a CCA instead of a CAA, it's not going to give us glutamine. It would give us proline. Okay, so this is totally different, and this would give us a different protein, which means that whatever wouldn't work right. Maybe it is an enzyme, maybe it's a hormone, whatever. It's not going to work right. Okay, you've got some, so this would be a missense. Now you've also got this thing called a nonsense mutation. Often this is going to give you a premature stop codon. Now remember, these codons are going to stop that segment of your amino acid sequence or that protein. Okay, so if your protein is not as long as it should be, if it's not right, then it is not going to work right. So you've got something that, will physically, come out wrong. Okay, so for example, if you get CAA and your, if this is your original codon CAA, and the tRNA for whatever reason brings a UAA instead of a CAA, it's going to tell that protein, stop. Instead of putting a glutamine on there, it's going to give you a stop. And so that means your protein's going to stop before it should. And if it's not as long as it should, it doesn't have the right amino acid sequence, well, we've made something that's not right. Okay, so these frameshift mutations, these are things that are very bad for us because it produces completely different protein sequences or amino acids, and it changes that reading frame to make the proteins that we need or the amino acids that we need. So let's say that the original was AUG, that means start, okay, and then it brings the codon, the original codon is supposed to say AAU for asparagine. Well, if for whatever reason your tRNA inserts an A right here, well, now instead of AAU, you got AAA. So see, it shifts the whole frame. So instead of having AAU for asparagine and then ACG, well, this U is shifted over here, and so you would get AU, AC, and then it would also shift this G, so it would be G, GC, and then you'd have a U floating around here somewhere. But now notice that we have completely different amino acids. So we've got AUG for start, but instead of asparagine, we get lysine. Instead of threonine, we get tyrosine. And instead of alanine, we get glycine. Okay, so this is a frameshift mutation. It means it takes that whole frame just because you inserted one, and it moves everything over. Again, gives us completely different amino acids, so we get total nonsense in the protein that is made, and that's not a good thing. Okay, you've got another assignment that you're going to do today. You're going to have a little quiz, and tomorrow you're going to have some other, work to do about mutagens. You're going to learn specifically about some types of disease. And so I'm going to try to put your work like this, where you work and then I kind of clarify what it is that you have done. If you have any questions, please, if you can, email me. That would be a very good thing. Or, under the classroom, do a chat. But if there's any questions, please do that. Don't panic if you don't understand something right away. We're going to get this, and there'll be little hiccups here and there. But I think we're going to enjoy this a lot. Have a very wonderful day, and I do miss y'all.